Investigating the structural and functional effects of mutations on phage proteins using cross-genome comparison. This was done by Sienna Denny and William Hadges. Phages, viruses that infect bacteria, act as naturally occurring models in genomics and protein studies due to their small genomes and overall simplicity. Phages have genomes that are dense in encoding genes, so they provide ample data for studying the relationship between genetic material and protein synthesis. Our investigation aimed to examine the relationship between genetic mutations and the resulting polypeptides in phages. The amino acids that result from any mutations are compared to determine changes in polarity. Polarity of the different amino acids have notable effect on the folding of a protein. We predicted that mutations affecting the first two base pairs in a codon will more often result in non-synonymous amino acid substitutions and significantly impact the structure of a protein, particularly in no known function proteins.
To briefly go over the modeling and computational methodology we used, we originally created a Python program that took two DNA sequences of the same length, compiled them by codon and amino acid, compared them directly to see if both sequences are the same, and then indexed all the differences. It outputted with the non-synonymous mutations, including which amino acids were substituted, returned all the transitions and all of the transversions.
The amino acid sequences of these gene sets were then modeled using the FIRE2 modeling program. This program produced predicted protein structures that were then visualized in PyMol based on secondary structure. Once the protein sets were visualized, the superalignment feature was used to superimpose the proteins over each other and record alignment scores for each set.
Our Python program outputted all the results that we need to directly compare the three gene sets that we investigated. Figure 4 is a comparison between the synonymous and non-synonymous mutations found in each of the three gene types investigated. Figure 5 is a comparison between the transition and transversion mutations found in each of the three gene types investigated as well. If you look closely at the charts, you can see that the known function gene, listed as NFK gene, had the largest number of total mutations, 7 mutations versus 1 for the tail chaperone and membrane protein genes, and had a good mix between synonymous and non-synonymous mutations and transition and transversions. This shows a more diverse mutational profile for that gene. Figure 6 gives us a deeper look into the non-synonymous mutations found, showing the original and replaced mutated amino acid. Surprisingly, only one of these major changes was caused by a transversion.
So to understand how these mutations affect protein structure, the gene sets were superimposed and aligned. Two metrics for how well the protein structures are aligned include the match-align score, which tells us a global alignment score based on how well the two matched up, as well as the root mean square deviation score, or RMSD, which tells us how far the final superimposed structure changed from the initial structure. When both of these scores are low, particularly when RMSD is below 1, this indicates a good alignment with high conservation. Draft 23 was compared to Pinhead Larry 22 and Figure 1, which have both been annotated as hypothetical no-known function proteins. This superimposition produced a match-align score of 53.895 and an RMSD of over 1, both of which indicate a relatively poor alignment. Next, Draft 25 was compared to Zelda 25, as shown in Figure 2, which have both been annotated as tail chaperones. And lastly, we looked at the alignment of Draft 15 with Schadenfreude 14 in Figure 3, which have both been annotated as membrane proteins. Both of these sets of superimposition scores had lower match-align scores and below 1 RMSD scores than Figure 1, suggesting better alignment than the set of hypothetical proteins and higher conservation between them. The no-known function proteins had the least conservation between them, as expected from the mutation analysis.
Our data generally matched what we expected. Based on the data collected, it seems to be the case that non-synonymous mutations are often caused by changes in the first two base pairs in a codon, as seen in Figure 5. And additionally, the hypothetical protein encoded by the unknown function gene had the least amount of conservation across the two genes compared, as shown by the highest RMSD score of all the compared genes. This can most likely be attributed to the larger number of non-synonymous mutations. The conservation between two tail chaperone genes was quite high, with a low match-align score and a low RMSD score as well. The distinct lack of non-synonymous mutations found is likely a reason for this conservation. This does lead to some interesting implications on a biological scale. As we look further into known function genes in the future, we might be able to look at the mutational profile of those genes to determine what their potential function might be. It could be assumed that known function genes that have been identified as having less diverse of a mutational profile might have more importance to the survival of the phage. This could be because during phage evolution, mutations would be selected against due to evolutionary pressure.